So understand and describe the gross anatomy of the hypothalamus and pituitary gland. Understand the regulation of the hypothalamic pituitary axis and the main hormones that are involved in this axis, including the feedback loop mechanisms. Describe and discuss the physiology and the biochemistry of the hypothalamic and pituitary hormones in general. What you're going to see through the module is that we're going to be looking at a specific axis in more details, i.e., we're going to be looking at the hypothalamic pituitary adrenal axis next week. Also, with Peter McCormick, you're going to be looking at the hypothalamic pituitary thyroid axis, and further on through the lectures with me, we're going to be looking at the hypothalamic pituitary and gonadal axis and fertility. So the lecture outline for today is that we're going to first cover the anatomy of the hypothalamus and the pituitary. We're going to give an overview of the hypothalamic pituitary axis, important hormones of the hypothalamus, the hormones of the anterior pituitary gland. We will mention all the hormones in the lectures that are coming, but we're going to make a special emphasis on prolactin and growth hormone, because in the lecture on Friday we will cover diseases which are important in prolactin and growth hormone secretions. Also, we're going to talk about the hormones of the posterior pituitary gland, such as antidiuretic hormone of ADH, also called as arginine vasopressin, and oxytocin or OT. So, just to start the lecture today, we're going to talk about the anatomy of both the hypothalamus and the pituitary. So, the hypothalamus is considered an endocrine master and servant. Why do we consider endocrine master and servant? Because the hypothalamus receives in the brain inputs from the outside and internal organs and homeostatic inputs, and tells the pituitary to secrete certain hormones. So the pituitary, which is here in red, is under control of the brain. So in this slide, you have the sagittal section of the brain, and the hypothalamus lies beneath of the thalamus. Hypo means under, and thalamus is the region of the brain here in blue. And it surrounds the lower portions of the third ventricle. If we look in the anatomy, what you can see is the cerebral cortex, in yellow the corpus callosum, the lateral ventricles, the thalamus, and the hypothalamus here in white, which is just above the pituitary gland. In the other side of the brain, we have the cerebellum, and we also have the part of the third ventricle. If you look at the sagittal section here, the eyes would be to your left, and the back of the brain will be to your right. If we look at this region of the hypothalamus in more detail, we can see that there is the optic chiasm and the pituitary gland. We're going to cover in more detail the anatomy of the pituitary gland. But first of all, we will study the anatomy of the hypothalamus. In gross manner, the hypothalamus is formed by the tuber cinereum or median eminence, and also several nuclei. The medial eminence is here just above the pituitary gland, and the hypothalamus, as you can see here in different colors, it's formed of different nuclei. What does it mean, nuclei? Nuclei are a group of hormones or of neurons that secrete certain hormones. These nuclei are not visible as an anatomical structure, but if you stain the brain for AVP, vasopressin, or oxytocin, or growth hormone releasing hormone, each nuclei has and contains some of the neurons that with axons which projects into the pituitary gland. We will cover this in more detail. So what you can see, guys, here is a 3D structure of the skull turning. And if we pay close attention, what we can see here in red is the pituitary gland, which is the size of a pea. This gland secretes a lot of multiple hormones that we will cover that regulate the homeostasis of the body, such as metabolism, reproduction, stress response, growth, lactation. Now this gland needs to produce hormones in response of stimuli or in response of homeostasis mechanisms that occur in the body, such as, for example, high glucose in the blood or high electrolytes in the blood, or regulates appetite or thirst, or, for example, fertility. So this is regulated by the other region that you can see here, which is just above the red gland, which is the hypothalamus, right? So the hypothalamus is where the axons and the neural content is produced and will control the anterior and posterior pituitary gland just underneath. So now that we know where it is located, let's study a little bit more in detail the gross anatomy. So the hypothalamus is a continuation of the tuber cinereum and the median eminence. Here you can see the median eminence. If you remember, this is attached to the hypothalamus and the hypothalamus to the brain. And it's attached to the pituitary through an expansion called the median eminence, or also pituitary stalk or infundibulum. And this joins the hypothalamus with the pituitary gland that you can see here. Next to it, you have the optic chiasm. And if we look at the pituitary gland is composed of two main lobes. One lobe is the anterior lobe or anterior pituitary. As we will study, this anterior lobe contains the hormone-producing cells. Or the posterior pituitary that does not contain the growth hormone-producing cells, but instead contains axons from the hypothalamus, and it expresses two types of hormones that go into the bloodstream, which we're going to study later on. The pituitary gland sits in the hypophyseal fossa or sella turcica or sphenoid bone. And as you can see here, there is the sphenoid bone and the anterior pituitary gland, posterior pituitary gland. What is important to note is that this gland is in intimate contact. However, embryologically, this gland comes from two different tissues. We will study this in this lecture a bit later. So the hypothalamus is very complex and is formed of multiple nuclei that contains specific neurons. Those are here written the different nuclei of the hypothalamus. So if we start, first one is the paraventricular nucleus, or also called PVN, and it contains ADH, hormone, which we will study later on, and oxytocin neurons. This ADH is arginine vasopressin or antidiuretic hormone, and it's important for osmolarity of the blood and retention of water. We will study this in a further lecture. Then we have the supraoptic nucleus, or SON, which also contains ADH neurons and also oxytocin neurons. Then we have the arcuate nucleus, which contains the dopamine and projections of the GnRH neurons or gonadotrophin releasing hormone neurons, which control reproduction. These neurons, actually, we studied today in the lecture, are not born in the hypothalamus, but they are born in the nasal placode and travel from the nasal placode all the way to the hypothalamus. Then we have the ventromedial nucleus, or VMH, that contains steroidogenic factor 1, which is a very important transcription factor for sex differentiation, also BDNF and POMC. We will cover this more in detail further on. And these neurons regulate stress response and reproduction and satiety. We also have the dorsomedial nucleus, DMH, which mediates leptin effects, and also we have the lateral and perifornical, LHA or PFA, which contains orexin, melanin concentrating hormone, and which control arousal and feeding. We will study some of these functions of these independent neurons in each nuclei. But you need to just know that each nuclei contains specific neurons, and we will cover some of them during the lecture. So this is a textbook image of how the hypothalamus looks like. If we look at it, right, we have the hypothalamus here above with the anterior pituitary below, the anterior gland, the posterior gland, just above the optic chiasm, and if we look from anterior to posterior, we can see that each nuclei is defined also depending on the position in the hypothalamus, right? So we have the lateral preoptic nucleus, medial preoptic nucleus, farther down we have the supraoptic nucleus, ventromedial nucleus, and all the way to the posterior nucleus. So basically it takes an anterior to posterior position, and remember that each specific nucleus contains specific hormones which will regulate the secretion of neuropeptides releasing hormones that control the pituitary gland. In this image, we have again the brain. In this case, the anterior part is to the right of the slide, with the posterior in the cerebellum. This is sagittal section. In this image, you have more clear where the nucleus are. So if we start for the anterior part of the brain, we have here the paraventricular nucleus in green. Then we have the lateral preoptic nucleus and medial preoptic nucleus, anterior hypothalamic, suprachiasmatic, supraoptic nuclei, and it's very nice picture here because you can see it in green and where the location is. In blue, we have the dorsomedial nucleus, ventromedial, and arcuate nucleus. And red, we have the posterior hypothalamic nucleus and mammary body. Remember that all these nucleus project axons into the medial eminence and will release factors that make the pituitary gland secrete certain hormones. We need to note that in the case of the anterior pituitary, these projections, axonal projections, do not touch the pituitary gland directly, but as you will study in few slides, the neurons secrete hormone-releasing hormones into a portal system of blood, vessels which are contacting the anterior pituitary gland. So the hypothalamus and the pituitary. The area of the brain directly underneath the hypothalamus is known as the pituitary gland. And we can see here in this draw the hypothalamus, and just underneath the infundibulum, which makes contact with the hypothalamus, and then we have the pituitary gland, which has two lobes, the anterior lobe and the posterior lobe. The pituitary gland also is known as hypophysis. The pituitary gland is connected to the hypothalamus through the infundibulum or pituitary stalk. And this is very important because, as you will see through the lectures, in the next 3 lectures, damage of the infundibulum, either by tumor growth, mechanical by accident, or through a disease, impairs the secretion of hypothalamic releasing hormones into the pituitary and totally breaks this axis. And this leads to severe endocrine disease. In here, what you can see is a sagittal section anterior to your left of the brain, the corpus callosum again, and we can see here very, very little the pituitary gland, which lies in the sella turcica. This, in your right, is an MRI image of a patient, healthy patient, showing the pituitary gland. You can see here very well the brain, the cortex, the corpus callosum, the cerebellum, and in this really bright spot, this is where the pituitary are. MRI is one of the line of diagnostics for pituitary and posterior pituitary defects. So we use MRI to detect whether the pituitary is smaller or hypomorphic or separated from the brain. So MRIs are very used in endocrine pituitary disease. This is the gross anatomy of the pituitary, human pituitary. And basically you can see here the connection with the stalk, and this is the anterior pituitary. What I would like to show the attention is that although this gland regulates reproduction, metabolism, homeostasis, electrolyte balance, stress, so many functions, it is the size of 1 centimeter, so the size of 1 pea. So has a very, very big job to do for the small size that it has. So now that we have talked about hypothalamus and the hypothalamic areas, right, and the different nucleus, what do we know about the anatomy of the pituitary gland? As I said, the pituitary gland sits in the sella turcica. Sella turcica comes from the Old Latin and comes from Turkish sitting. Can you see here the sitting of the horse? And it has this shape, right? That's why it's called the bone sella turcica. The pituitary gland is situated below the brain and is encased in this bony cavity called sella turcica or Turkish saddle, also known as sphenoid bone. It's the size of a pea and is known as the master regulation of endocrine glands. And the reason is because the pituitary gland secretes hormones that will control loads and loads and loads of different organs, which in turn will secrete more hormones which will feed back and give information to the brain. We will cover in the next lectures which organs are these ones, but just to give you a brief introduction, it regulates gonads, such the ovaries and the testes, it regulates the adrenal, it regulates pancreas, it regulates the bones, it regulates the breast, and it regulates growth, right? And it also targets the liver. So you will see through the lectures and understand each axis that the pituitary controls. So the anterior and the posterior pituitary that you can see here are also called neurohypophyses or adenohypophyses. The anterior pituitary gland contains hormones and does not contain axons, and it is the true endocrine secretin. So that's why it's called adenohypophysis. The posterior part is an extension of the brain, contains axons from the hypothalamus and terminal nerves that will secrete two hormones, oxytocin and vasopressin or antidiuretic hormone. But the anterior pituitary does not contain axons, and it comes from another tissue which we will study today. The posterior is called also neurohypophysis, and the anterior adenohypophysis. They effectively, although form one unique gland, work separately under the regulation of hypothalamic inputs. In this cartoon, we have more of the hypothalamic pituitary regulatory system. And as you can see here, we have the anterior pituitary gland. What you can see here is the portal vessels and the posterior pituitary gland. As I said, both the anterior and the posterior are connected to hypothalamic inputs. What you can see here is that there are neurons. These neurons project axons into the artery or portal system of veins. We have the suprahypophyseal artery that goes into the brain, into the medial eminence, collects the hormones, takes them into long portal vessels, and will take these hormones, peptides, or releasing factors into the anterior pituitary. The posterior pituitary, the axons actually grow into the posterior pituitary and also connect to the veins, inferior hypophyseal artery, and directly contact them, releasing all the hormones. So now that we know about the gross anatomy of the pituitary gland, we're going to study the development of this pituitary. The development of the pituitary is controlled genetically, and it's very important to understand the latest physiology. So it starts, we have here in A, the Rathke pouch or oral ectoderm, which touches the infundibulum or the brain. There is a successive invagination of the Rathke pouch that touches the infundibulum. Then the oral ectoderm detaches of the oral cavity and forms this pouch. This pouch that comes from the oral ectoderm is in contact with the posterior part or the infundibulum. The infundibulum here in yellow will come from the CNS part or the nervous system. So the infundibulum and Rathke pouch develop from the oral ectoderm and the posterior lobe from the neural ectoderm respectively. So that's why it has double tissue embryonic origin. So remember, infundibulum or posterior lobe comes from the CNS, and the Radcliffe pouch or anterior pituitary from the oral ectoderm. So again, the different mechanisms is that the Radcliffe pouch constricts at the base. The Radcliffe pouch completely separates from the oral epithelium. The final pituitary gland is formed of the pars distalis, pars tuberalis, and pars intermedia in humans, not in other species. The neurohypophysis is formed by the development of the pars nervosa, infundibulum, and stem and median eminence. And we will cover this a little bit more in detail in the next slides. So, what we use to study the development of the hypothalamus and the pituitary gland is the mouse as a model. The mouse embryogenesis is very similar to the human embryogenesis. And we've been using this tool to study the genes that control the development of the pituitary gland. What you can see in this slide is a sagittal section of a mouse embryo at embryonic day 8.5. You can see here in red the oral ectoderm, and in gray will be the brain. So the oral ectoderm starts thickening and will form the Radcliffe pouch here in red. The interaction between the Radcliffe pouch and the above infundibulum will form the embryonic hypothalamic pituitary axis. The Radcliffe pouch will start expressing transcription factors that will tell these cells to become pituitary gland. And the infundibulum will start also expressing transcription factors that will tell this tissue to become posterior part of the pituitary. At this stage, both structures are formed of stem cells. A little bit later during development at 14.5, you can see here in red the Radcliffe pouch evaginates and form a definitive pouch in red. This will form the anterior pituitary that basically contains all the hormone producing cells. The posterior lobe that comes from the CNS or infundibulum, as you can see, comes from the nervous tissue and will contain the axons from the hypothalamus, but not the main hormone producing cells. And finally, at later stage or almost at birth, we can see the three lobes of the pituitary gland. Okay? The three lobes of the pituitary gland are the posterior lobe, as we said, in yellow, which contains the CNS and the axons, and the anterior in red that contains the intermediate lobe and anterior lobe. Now, using mouse transgenics and mouse genetics, as well as zebrafish and human genetics, we have identified that this process of development is tightly regulated genetically, and mutations in genes that control this development leads to abnormalities of the pituitary gland, which are causative of human disease, and we're going to cover a couple of them. This is a draw from one of my articles. I really recommend printing. I don't know if you have access to these PDFs, but otherwise I can ask and upload it in QM+. But this draw that my student did depicts the development steps of the pituitary. So we have at early stage, those are sagittal sections. This is the ventral diencephalon or the brain. Underneath has the Radcliffe pouch with green cells. These green cells are stem cells. Then this pouch evaginates and forms a definitive pouch, and then forms at E13 embryonic day this structure which contains the posterior part of the pituitary with the green cells that contain the stem cells of the future pituitary. In yellow, we have the committed cells, so the stem cells start cell lineage determination and undergo differentiation. And you can see here the posterior lobe, the marginal zone, which is the stem cell niche, and the cells start differentiating and forming the growth hormone producing cells, which you have here in orange. The future pituitary gland at birth contains the three lobes. The posterior, remember, contains the axons, with the intermediate lobe, the marginal zone that contains the stem cell population of the pituitary gland, and the terminally differentiated cells, which contains the hormones. If you would like more information on the genes and the development, there is in this review, it's very self-explained. So you don't need, guys, to understand this, right? Only 3 genes are important. But this is just to depict, as I've shown you before, this is the Rathke pouch, right, that will become the future pituitary. Our laboratory and other laboratories have shown that the genes and transcription factors involved in Rathke pouch specification and terminal differentiation is quite complex. So there are lots of homeobox transcription factors such as HESX1, TBX, 6-3, DRAX, which are important in the brain to give signals to the Rathke pouch. Then we have genes which are important for stemness, like for example SOX2, PTX, Win factors, and GLIS, which are important in the stem cells to trigger a cascade of differentiation factors. These genes will tell these cells to stop being progenitors and stem cells and to start differentiation, and will take 3 different lineages which will give the future hormone producing cells. So the main genes that you need to know, it's the TPID or TBX9, SF1 transcription factor, and PID1. The cells that express the TPID will undergo further differentiation into 2 lineages, producing ACTH or adrenocorticotrophin hormone and also MSH. So this is one lineage. Mutations in humans with TPID don't produce ACTH, and these kids are very ill, and we will cover this further into the lecture in another session. You just need to remember 3 lineages: TPID1, SF1. SF1 will regulate the reproduction through the terminal differentiation of GnRH of gonadotrophin releasing hormone and produce luteinizing hormone and follicle stimulated hormone, which will impact on the gonads. It will also have, through GATA2, part of the thyroid stimulated hormone. The other important transcription factor is PID1. PID1 will have 3 different lineages or 3 different terminal differentiation hormones: TSH, thyroid stimulated hormone, growth hormone, and prolactin. TSH is important for thyroid, growth hormone for growth, and prolactin for lactation. So every year more genes are added to this genetic cascade as we identified important genes important in the regulation of the differentiation of each specific cell type. Indeed, my laboratory has identified beta-catenin as an important regulatory gene for the PID1 lineage, TCF, and also HESX1 and 6-3. So what happens when mutations in these transcription factors occur that impair the production of hormones? One example is this. So this is a patient, has 8 years old, and his height is 84 centimeters. So as you can see, has severe developmental delay and has severe hypothyroidism. This is a patient from my collaborator, Professor Mehudatani, working in Great Ormond Street. This patient was diagnosed with hypopituitarism, and if you look here at the MRI, again you can see the cortex, the corpus callosum, but if you look at the bright spot, it's very small, indicating a small pituitary gland and hypopituitarism. So this patient has a mutation in PID1 transcription factor. So if we go back into the cascade, PID1 is at this level. So that means that PID1 does not produce TSH, growth hormone, and prolactin. So this patient had absence thyroid stimulating hormone, growth hormone, that's why it's dwarf, and prolactin. So this is just to show you the importance of these genes in human disease. But equally, mutations in these transcription factors can cause other severe disease, which we're going to study in the following lectures. And what you have here is the shortest man on earth that was from Nepal and has congenital hypopituitarism, so no hormones are secreted from the pituitary gland, and has severe dwarfism. But the opposite spectrum, we have a giant, a man that has grown too much because has too much hormone in the pituitary due to a pituitary tumor. So what we will also study is why mutations in these transcription factors or molecules that are important in the regulation of the pituitary gland can give us this type of conditions. As I have told you, for example, the transcription factor beta-catenin is very important in the stem cells of the pituitary gland. And mutations in beta-catenin, we have shown in my laboratory, produce pituitary tumors. You can see here in the right an MRI with the brain, the cortex, corpus callosum, and you can see a big tumoral mass where the pituitary gland should be, compared to a healthy individual that has just one bright spot where the pituitary is. These tumors of the pituitary gland lead to severe endocrine disruption with lack of multiple hormones, but also affect the hypothalamus, and the hypothalamus regulates feeding behavior. So we have here a picture of a patient with one of these tumors, craniopharyngioma, after operation, and what you can see, the patient was before operation, before the tumor, and after the tumor had been resected, it leads to severe comorbidities of hypothalamic disease. So basically, severe obesity because the hypothalamic area was affected. So basically, what I'm trying to picture here is how important is not only for normal homeostasis, but how mutations in some genes and the regulation of the HP axis can lead to severe abnormalities and disease of the metabolism, growth, and also tumor formation. On the lecture on Friday, we will cover the tumor parts of the pituitary. So, As you know, the pituitary gland, the main function is to release hormones into the bloodstream. So the pituitary needs to be highly vascularized. So the blood supply of the pituitary comes by two blood vessels, two branches of the internal carotid. So it has the superior hypophyseal supply and the inferior hypophyseal supply. You can see here with the arrow the superior hypophyseal, that it basically goes into the medial eminence and into the here the infundibulum. The inferior hypophyseal goes into the neurohypophysis or the posterior gland. Remember here we have in orange the adenohypophysis and in yellow the posterior pituitary. What is important, if you want to be an organ that needs to secrete lots of hormones, it needs to be highly vascularized. In the case of the hypothalamus connection neurons to the anterior pituitary, right, is through a portal system of vessels, you can see here in blue, that are connected between the hypothalamus and the pituitary. It forms two plexus. You can see here the secondary plexus or hypophyseal, also known as hypophyseal portal system. So the superior hypophyseal supplies the median eminence, as you can see here in the draw, and the upper part of the stalk. The inferior hypophyseal supplies the lower part of the stalk and the neurohypophysis or the posterior pituitary. The arteries supply the medial eminence and the stalk and as a capillary plexus. So you can see here in blue little capillary plexus. So the endocrine cells will secrete their hormones into this capillary plexus that will drain and go into the blood system and target peripheral organs. This is a more intricate yet more detailed anatomy of the hypothalamus, the axonal tracts, and also the blood vessel system. We're not going to cover everything, but it's for you to have and to have a look while you're studying. But basically, in a gross way, we have the anterior pituitary, the posterior pituitary. We can see the two main supply of blood, superior hypophyseal artery that branch intracranial portion of the interior carotid artery and supplies blood to the median eminence and higher part of the infundibulum or stalk, right? What you can see here also are the nuclei and the axonal projections, right? The axonal tracts coming from the different nuclei of the hypothalamus, such as the supraoptic, the paraventricular, and these ones, axons, will extend. In one case, if they release hormones for the pituitary gland, will have contact with the um, portal system of blood vessels, which will drain into the anterior pituitary. In the case of the posterior, this doesn't drain into portal system, but it touches the hormones are produced, the peptides are produced in the bodies, travel through the axons, and are kept in the posterior pituitary waiting for its release. And there are also only two types of hormones at this stage. One is oxytocin and vasopressin. We will study the function of these ones in further lecture. So let's go over again the pituitary veins. Obviously, if the importance of the organ is to supply hormones to target organs, these hormones need to be circulating into the mainstream and needs to be released into a system of veins. So there are two capillary plexus, as I've said. One is here superior, hypophyseal portal vein that contacts the hypophyseal portal veins, the hypothalamus with the anterior pituitary. The capillary plexus is in median eminence and stalk are drained by portal veins. These veins pass to the anterior lobe of the pituitary and veins pass to a secondary plexus which is found in the anterior pituitary. This provides a route for neurosecretory substances, which we will study, to release from the hypothalamus and pass into the anterior lobe of the pituitary, which in turn will activate cell types to release some hormones. So this is also for your references, and again is a bit more intricate, but you can keep it for obviously your studies, but it actually shows a draw of everything we have covered in this first lecture, which is the hypothalamic nuclei. So again, the hypothalamic nucleus from anterior to posterior have different names. All you need to remember is that each nuclei contains neurons that are specialized in certain production of neuropeptides that will activate the pituitary gland. And these are in different locations like preoptic nuclei, right? The arcuate nucleus, the suprachiasmatic nuclei, paraventricular nucleus, dorsomedial nucleus, the posterior, ventromedial, supraoptic, and arcuate nucleus. And each one contains certain population of neurons, which we will study. These neurons project in two ways. Once into the anterior pituitary, but they don't make contact with the endocrine cells. No. What they do is that they secrete their factors in the plexus, capillary plexus, which is drain all these peptides and hormones to the anterior pituitary, which contain the troph cells or trophic cells, which will produce growth hormone, thyroid stimulating hormone, adenocorticotrophin hormone, follicle stimulated hormone, so on and so forth. The posterior doesn't drain into the portal system, but it directly touches into the posterior lobe, which will secrete only two types of hormones, oxytocin and vasopressin. So, the importance of the hypothalamic adenoapical portal system. What is important to remember is that is a vascular link between the median eminence and hypothalamus and the anterior pituitary. Is called hypophyseal portal system. Now, if you imagine, for example, a car accident or a tumor growing in this region, that will break the connection between the hypothalamus and the pituitary gland. So the neurons will not be able to actually tell the pituitary gland what they need to secrete. The releasing hormones, which we will study in next lecture, are secreted by the hypothalamus into the hypothalamic adenohypophyseal portal vessels, which are here in blue, and these are carried into the anterior pituitary. The releasing hormones do not enter the hypothalamic releasing hormones do not enter the systemic circulation. They go into the anterior pituitary, and the anterior pituitary will secrete hormones into the circulation. the anterior pituitary gland does not contain neurons or hypothalamic inputs. So the hypothalamic inputs stop in the plexus in the hypophyseal portal system. So, guys, we're going to have a break, right? And then we will cover the next lecture. We will go into an overview of the hypothalamic pituitary axis, and we're going to cover mainly the hormones of the hypothalamus and how these ones control the pituitary gland. So I think we have 30 minutes break, so we can be back here in 30 minutes and we will restart the lecture.